Okay. Today we are going to discuss about oxygen dissociation, oxyhemoglobin dissociation curve. Oxyhemoglobin dissociation curve. Oxyhemoglobin dissociation curve. Dissociation curve. This is very most important for NEET purpose, Nana. From this topic, the questions will come differently, and most of the children are going to get the confusion to answer the questions which will come from this area. If you listen carefully, you are able to attempt the question perfectly properly. It's very easy to attempt the questions. That's why listen carefully. Oxyhemoglobin dissociation curve. Now we have to discuss about oxyhemoglobin dissociation curve. What is oxyhemoglobin dissociation curve? Here you can take the relation between percentage saturation of hemoglobin. The relation between percentage saturation of hemoglobin. Percentage saturation of hemoglobin. Percentage saturation of hemoglobin. The relation between percentage saturation of hemoglobin and partial pressure of oxygen. Partial pressure of oxygen. Partial pressure of oxygen is called as oxyhemoglobin dissociation curve. If you will take this oxyhemoglobin dissociation curve, the relation between percentage saturation of hemoglobin and partial pressure of oxygen is called as oxyhemoglobin dissociation curve, and it's a sigmoid curve. It is a sigmoid curve. It is a sigmoid curve. Sigmoid curve containing a S shape. That's why it is called as a sigmoid curve. To develop, to bind the oxygen with hemoglobin, to bind oxygen with hemoglobin, to bind oxygen with hemoglobin, oxygen with hemoglobin. Some factors are involving to bind the oxygen with hemoglobin. Some factors are involving. We are going to discuss about those factors. Which factors are involving? These factors are involving in the binding of oxygen with hemoglobin. If we will take that factors, those factors are PCO2, partial pressure of carbon dioxide, and hydrogen ion concentration, hydrogen ion concentration, and temperature. These factors are involving in the binding of oxygen with hemoglobin. These factors are PCO2, partial pressure of carbon dioxide, and the hydrogen ion concentration, temperature. These three factors are involving in the binding of oxygen with hemoglobin. When oxygen binding with hemoglobin, due to the binding of oxygen with hemoglobin, oxyhemoglobin developing. This oxyhemoglobin again, it must be which dissociate. The dissociation of oxygen with hemoglobin is called as oxyhemoglobin dissociation curve. How it is dissociating, we have to see. And here you can take the binding of oxygen with hemoglobin. We have to discuss with a which is on a table. Here you can take the binding of oxygen with the hemoglobin. How it is binding? We have to see this. The binding of oxygen with the hemoglobin. It's the x-axis and y-axis. Above the x-axis, this is the oxygen O2. And here you can take its CO2. The percentage of oxygen 10 mmHg, 20 mmHg, 30 mmHg, 40 mmHg, 50 mmHg, and 60 mmHg. And here you can take carbon dioxide. Percentage of carbon dioxide 10 mmHg, 20 mmHg, 30 mmHg, 40 mmHg, 50 mmHg, and it's 60 mmHg. When oxygen is more, partial pressure of oxygen is more, here the hemoglobin combines with oxygen and a line is developing, a curve is developing. It looks like as a S shape, that's why it is called as a sigmoid curve. This is called as a sigmoid curve. If oxygen is more, the curve will develop to left side. This is called as a left side, left, and it's right side. In the left and right side, most of the persons are going to get the confusion which is left and which is right. That's why here you can take, if you will stand as like as this is the left side and this is the right side. We have to see that only. That's why here you can take it's a left side and this is the right side. If more oxygen is present, the oxyhemoglobin curve moves towards left side. From this area, the questions will come. In the need, they have to ask in tissues, less oxygen is present, less O2 is present, then oxyhemoglobin dissociation curve, then oxyhemoglobin dissociation curve, oxyhemoglobin dissociation curve. Here you can take first option, moves towards left side. Here you can take second one, moves towards right side. Third option, here you can take third option, the curve does not change. The curve does not change. Curve does not change. Fourth option, none. Here you can take, just now we were discussed, if more oxygen is present, the oxyhemoglobin curve moves toward left. Why? Because it is taking the hemoglobin taking more oxygen and it must move towards left side. But here the question is in tissues less O2 is present. If less O2 is present, the oxyhemoglobin dissociation curve moves towards right side. Moves towards right side. Here you can take our option moves towards right side. Why? Because here they gave in tissues less O2 is present, then the oxyhemoglobin dissociation curve less O2 is present. That's why from oxyhemoglobin, it must breaks into oxygen plus hemoglobin. Oxygen plus hemoglobin. It must breaks into oxygen plus hemoglobin. When oxygen is detaching from the hemoglobin, it must move towards which side, Nana? Right side. So from this area, the questions will come as like this. Most of the persons are going to get the confusion here only. They won't answer perfectly. They have to confuse. That's why one thing you have to remember: if more oxygen is present, the oxyhemoglobin dissociation curve moves towards the left. If less oxygen is present at the tissues, it must be moves towards the right side. Why? Because if the tissues are containing less oxygen, from oxyhemoglobin, the oxygen releasing, oxygen detaching from the hemoglobin, and this oxygen enters into the tissues. That's why the curve moves towards the right side. This is about the oxygen dissociation curve. If the tissues are containing less O2, the oxyhemoglobin dissociation curve moves towards right side. Just now you are discussed to bind the oxygen with hemoglobin. Which factors are involving? 3 factors are involving. They are the carbon dioxide, hydrogen ion concentration, and temperature. These are the 3 factors which are involving in the binding of oxygen with hemoglobin. How they are involving? We have to see. If more PO2 is present, more partial pressure of oxygen is present. If more partial pressure of oxygen is present, when more partial pressure of oxygen is present, then less PCO2 present, less concentration of carbon dioxide present. Along with this less concentration of carbon dioxide, less temperature is present. Along with the less temperature, hydrogen ion concentration also less. Less hydrogen ion concentration is present. Less H plus concentration is present. Less H plus concentration is present. At this time, the oxygen binds with hemoglobin. When the oxygen binding with hemoglobin, the curve moves towards which side, Nana? Left side. If oxygen binding with hemoglobin, the curve moves towards with left side. When the oxygen binding with hemoglobin, when more oxygen is present, when more partial pressure of oxygen is present, at the time only the oxygen binds with hemoglobin. When oxygen binding with hemoglobin, the curve moves towards left side. From this area, the question will ask as like as this: if more PO2 is present, the curve moves towards first option moves towards left side, second option moves towards right side, third option the curve does not change, fourth option none of these. What is the answer now?
First option, sir. First option, sir. Moves towards left side.
Good, very good, excellent. Moves towards left side. Why? Because more oxygen is present. When more oxygen is present, the oxygen combines with hemoglobin. When the oxygen combines with hemoglobin, here the curve moves towards the left side. If more PO2 is present, if more PO2 is present, partial pressure of oxygen is present, then first option, high CO2 is present, high PCO2 present, high PCO2 present. Second option, less PCO2 present, less PCO2 present. Third option, no change in PCO2, no change in PCO2. Next, fourth option, none. Fourth option, none. What is the option, none? If more PO2 is present, if more PO2 is present, then high CO2 present, less CO2 present, no change in CO2, PCO2, none. What is the option?
Less second option, sir.
Good, very good, excellent, Nana. Very good, you are following very well. Excellent, Nana. Less CO2 is present. If more oxygen is present, less CO2 is present. When more oxygen is present, the oxygen binds with hemoglobin. When oxygen binds with hemoglobin, the curve moves towards left side. Why? Because here you can take when it is moving towards left side, the percentage of oxygen is increasing. Here only 0, 10, 10 mmHg, 20 mmHg, 30 mmHg, 40 mmHg, 60 mmHg. If the curve moving towards this side, x-axis, this is the left side, more oxygen is present. If the curve move towards y-axis, here you can take the curve moving towards y-axis. Here means what, Nana? Less oxygen is present here. You can observe that, but more carbon dioxide is present here. If less oxygen is present, here I would like to give one more. Here, if less oxygen is present, if less O2 present, if less O2 present, at this time, more PCO2 present. When more PCO2 present, here more temperature present, more temperature present, more temperature present. When more temperature present, here more hydrogen ion concentration is present. More hydrogen ion concentration is present. More hydrogen ion concentration is present. When less O2 present, when less O2 present, at that time more PCO2 present, more temperature present, more hydrogen ion concentration present. If more temperature present or more PCO2 present, more hydrogen ion concentration present, the oxyhemoglobin dissociation curve moves towards right side. The oxyhemoglobin dissociation curve moves towards which side, Nana? Right side. When less O2 is present, if less O2 is present, definitely it must be more carbon dioxide, more PCO2, more partial pressure of carbon dioxide. If more partial pressure of carbon dioxide is present, definitely more temperature present. If more temperature present, definitely more hydrogen ion concentration present. If more PCO2 present, definitely it must be more temperature and more hydrogen ion concentration. When more PCO2, more temperature, more hydrogen ion concentration is present, at that time the oxyhemoglobin dissociation curve moves towards y-axis. At that time the oxyhemoglobin dissociation curve moves towards y-axis or moves towards the right side. If you say the right side, which axis is present? Y-axis is present. It must move towards y-axis or right side. This is about oxyhemoglobin dissociation curve. the oxyhemoglobin dissociation curve depends upon the oxyhemoglobin dissociation curve depends upon PO2, PCO2, temperature, and hydrogen ion concentration. There must be a condition. If more PO2 is present, definitely less PO2 present, less temperature present, less hydrogen ion concentration present. When less temperature present, less hydrogen ion concentration present, less PCO2 present, at that time the oxyhemoglobin dissociation curve moves towards x-axis. This x moves towards x-axis or moves towards left side. One more condition also present. If less oxygen is present, definitely more PCO2 present, more PCO2 present, more temperature present, more H plus ion concentration present. If more H plus ion concentration, if more temperature, if more PCO2 present, this oxyhemoglobin dissociation curve moves towards right side. In these conditions, how the oxyhemoglobin dissociation curve moving to right side and left side, we have to know perfectly properly. From this area, the NEET questions will come. Differently, the NEET questions will come. If you know this subject, each and every question you are able to attempt that. That's why listen carefully. This about what Nana oxyhemoglobin dissociation curve. And I would like to give the definition once again. Listen to me. The relation between percentage saturation of hemoglobin, the relation between percentage saturation of hemoglobin and partial pressure of oxygen is called as oxyhemoglobin curve. It's a sigmoid curve. It is containing a somewhat S shape. That's why it is called as a sigmoid curve. To develop, to bind the oxygen with hemoglobin, some factors are involving. These are the factors which are involving in the binding of oxygen with hemoglobin. They are PCO2, H plus ion concentration and temperature. And in some condition, how the oxyhemoglobin dissociation curve moves towards left and right. Just now we were discussed. This is about oxyhemoglobin dissociation curve. And now we have to discuss about Bohr effect. Bohr effect. Children followed. Anybody want to once again, Nana?
Sir,
Tell me, Nana. Any doubt, Nana?
sir, we taken if more PO2 is present, sir. In this, if less O2 only present, sir, it is not a PO2.
If more PO2 means more partial pressure of oxygen. When more partial pressure of oxygen is present, more oxygen is present. When more oxygen is present, definitely less PCO2 present. If more oxygen is present, there is no choice to present of carbon dioxide more amount. If more oxygen is present, definitely, absolutely, less carbon dioxide is present, less temperature is present, less hydrogen ion concentration is present. If more PCO2 present, definitely more temperature present, more hydrogen ion concentration present, definitely 100% less O2 present. There is no choice to present more oxygen and more carbon dioxide. Definitely one of the gas, definitely it must be more than other gas. Okay, Nana?
Okay, sir.
Sir, what is the full form of mmHg, sir?
Millimeters Hg, the partial pressure of oxygen, how much oxygen present in the air, that must be indicated with mmHg. Okay, Nana?
Yes.
Okay, now we have to discuss about Bohr effect. Bohr effect. Bohr effect. It's a simple definition. We have to discuss about the definition. Listen to me. Bohr effect. Bohr effect. Bohr effect. Bohr effect. The effect of PCO2, the effect of the effect of PCO2 and H plus ion concentration. H plus ion concentration on oxygen affinity. Oxygen affinity is called as Bohr's effect or Bohr effect. It was discovered by the Bohr scientist. That's why it is called as the Bohr effect. Just now we were
Sir,
discussing.
oxygen affinity of hemoglobin.
Oxygen affinity of hemoglobin or the affinity of oxygen. If more oxygen is present, definitely less PCO2 is present, less hydrogen concentration is present. It has defined as a definition by the Bohr scientist. That's why it is called as a Bohr effect. Just now we were discussed the effect of PCO2, the effect of partial pressure of carbon dioxide, and H plus ion concentration on the oxygen affinity of hemoglobin or oxygen affinity is called as Bohr effect. If more PCO2 present or more H plus ion concentration is present, definitely less oxygen is present, and it was discovered by Bohr scientists. That's why it is called as the Bohr effect. This is about the Bohr effect. The effect of PCO2, and H plus ion concentration on oxygen affinity on PO2 on oxygen affinity or PO2 is called as a Bohr effect. This is about the Bohr effect, and this is called as oxyhemoglobin dissociation curve and Bohr effect. Oxyhemoglobin dissociation curve and Bohr effect. And here in previous class we were discussed the hemoglobin able to take 4 molecules of oxygen. In previous class we were discussed the hemoglobin able to take 4 molecules of oxygen. Hemoglobin containing one ferrous molecule or iron molecule. This able to take 4 molecules of oxygen. When hemoglobin able to take 4 molecules of oxygen, it must satisfy 100%. The capacity of hemoglobin to take 4 molecules of oxygen. When it is taking 4 molecules of oxygen, it cent percentage it must be satisfied. How it is satisfied we have to discuss. Yes, at 100 mmHg, at 100 mmHg of PO2, if the partial pressure of oxygen more at 100 mmHg of PO2, 97% of hemoglobin satisfied. HB means hemoglobin. 97% of hemoglobin satisfied. Means 97 means 100%. At 100 mmHg PO2, when partial pressure of oxygen is more, the hemoglobin satisfied 100%, probably 97%, means 100%. The hemoglobin, 97% of hemoglobin is going to satisfied. At resting position, at resting stage, what is meant by resting stage, Nana? If it does not do any work, if we simply sit in a chair, if we simply sit, that is called as a resting position. At resting position, at 40 mmHg, of PO2, at 40 mmHg of PO2, 75% of hemoglobin satisfied. 75% of hemoglobin satisfied. that when we are in resting position, 75% of hemoglobin satisfied. What is the difference between 97% and 75, Nana? The difference is only 22%. When we are in resting position,
2 minutes left.
when we are in resting position, we must be utilize only 22% of oxygen. Utilize only 22% of O2. At the resting position, we have to utilize only 22% of O2. The 1/5 percentage of O2 we have to utilize. 1/5 part of O2 we have to utilize. Remaining 4/5 part present in part of O2 present in hemoglobin. At the resting position, we are utilizing very less quantity of oxygen. Only 22% of oxygen we are utilizing. 1/5% of oxygen only we are utilizing. In the resting position, our hemoglobin, our blood containing 4/5 part of hemoglobin. The more hemoglobin present in our blood at the resting position. But when the person do work, when the person walk speedily, when the person runs speedily, when the person will do the exercise, when the person will do the hard work, at that time, the hemoglobin release more percentage of oxygen to the cells.